So, hi everybody. Welcome to our 6th COVID Community Town Hall, sponsored by the Stanford School of Medicine Office of Community Engagement and the Department of Epidemiology and Population Health. My name is Lisa Goldman Rosas. I'm assistant professor in the Department of Epidemiology and Population Health, and I get to direct our School of Medicine Office of Community Engagement, and I am so glad to be your moderator today. So why don't you go to the next slide, Maria? All right. Today's town hall is focused on vaccine trials, and really there has never been a better time to discuss this topic. So I'm so glad you're here. As you've heard me say on previous town halls, staying informed is a great way to manage our anxiety and get through this pandemic. So be sure to listen carefully, ask lots of questions, and share what you learn with your friends. All right, so before we get started, oh, you can go back, Maria, sorry. We have a few housekeeping items. Okay, so first, if you would like interpretation in Spanish and Espanol, please click the link below and you can hear the simultaneous translation in Spanish. And I should have also said, bienvenidos a todos y todas que nos están escuchando en español. Qué bueno que nos hayan acompañado el día de hoy. All right, and we have Claudia and Esperanza in the interpretation room who are interpreting for you. And so Claudia and Esperanza, go ahead and say hello. All right, and second, many of you submitted questions ahead of time, and we will try to get to as many of those questions as possible. If you'd like to submit questions during the event, and we really hope that you do, go ahead and use that question and answer button at the bottom of your screen. Don't use the chat button because we are monitoring that question and answer button. We will also try to get to as many of those questions as possible. También, si quieren poner sus preguntas en español, adelante. In addition, today we are very honored to have a Stanford physician, Dr. Khosravi. I don't know if you can say hello on your screen, who is with us, and he will also be monitoring the question and answering as many questions as he can get to. That's one of the ways we are trying to get to more of your questions. So if you have a personal medical question, ask your doctor, but Dr. Khosravi will try to answer as many questions as he can. All right, you can also give a thumbs up icon to questions that you like that are put in the question and answer button, so you don't have to answer put questions twice. Okay, and one last note is our next community town hall will be in January. We are taking a break in December, and in January it will be on vaccine readiness, so a direct follow-up of this town hall. So be sure to mark your calendars for that one. Okay, I think we've gotten that all out of the way. So now I get to introduce our panelists. We have a wonderful lineup of panelists today. I think you're going to really enjoy it. So first we have Dr. Stephen Goodman. He is a professor of epidemiology and population health here in our School of Medicine and an epidemiologist. We have Dr. Philip Grant. He is a physician and infectious disease physician, also here in Stanford School of Medicine. And we have Dr. Vivian Levy. She is the chief of infectious disease in San Mateo Medical Center, as well as an adjunct faculty member here. Last, but most certainly not least, we have Dr. Chaik Dubeni. He is a professor of family medicine and directs the Center for Health Equity and Community Engagement at the Mayo Clinic Comprehensive Cancer Center. And Dr. Dubeni is joining us from out of town. You can see he's not here from our local area, so we want to welcome him to the neighborhood. We are so glad that he is able to join us. Okay. So if you've listened to any of these town halls before, you know that I like to ask each of the panelists to share something personal before they get started so we can get to know them a little bit better. Today, since we're talking about vaccine trials, I thought what we could do is each panelist can think of a research study that you've participated in, and you can just share a brief tidbit of what it was like to be a research participant for you. And as always, I will go first. So it gives you, I see Dr. Goodman's thinking of his, it could be you or your child perhaps. I'll share one that I did. I have participated in a lot of studies actually, and I was thinking of one that I participated in when I was pregnant because I had to stop participating midway through. As a researcher, I have written hundreds of consent forms, so I know you're allowed to say you want to stop participating at any time. And this was a fetal MRI study, and I think I was maybe 8 months pregnant and they put me in the MRI tube and I couldn't see out. So I started shouting to the MRI technician to take me out, take me out. I wanted to stop participating. So that was the end of my participation in that study. I don't remember if I got the gift card or not. Maybe I didn't because I didn't finish the study. Okay, so each of us have experienced what it's like to be both a research participant and a researcher. And since we're going to be talking about research today, it's good to remember both sides. All right, so let's move to our first panelist without any ado. And remember, each of the panelists is going to share their slides or share their presentation, and we will do the question and answer when they're finished. So go ahead and submit your questions as they go, and then we'll get to them afterwards. All right, Dr. Goodman.
Thank you so much, Lisa. And hello to everybody who's here today. A trial I've been in, actually, I've never been that sick, so I haven't been in a therapeutic trial, but the one that actually I remember most is a trial that I was enrolled in that I didn't know I was a part of. I hadn't been asked for consent. I was part of some sort of test, and I found out afterwards. And here I am, like 20 years later, still angry about it. So it shows the importance of consent, even for things that are very benign, and being completely informed when you're in a study of any kind. This was just a survey study. But the topic for today, I don't know exactly when it was chosen, but it was many, many weeks before we knew that we were going to get this avalanche of information about vaccines just this week. And that's what I'll be talking about today, and some of the other panelists. So let's just jump right into it. Next slide. So this is from the New York Times just yesterday. They keep a coronavirus vaccine tracker, and they tell us how many trials, how many vaccines are in various phases of testing. Right now, there's a total, they say, of 54 vaccines that are being tested. 38 are in phase 1. You'll see the numbers don't quite add up to that. I don't know why they're reporting that. But phase 1 is just very preliminary safety testing in human beings. Phase 2, where they're reporting 17, is when the populations are expanded. Phase 1 might involve 30 or 50 people. Phase 2 might be 200 or 400, where we're looking at both safety, but also immune response. Phase 3 is what you've been hearing in the news, and we'll talk most about, which are the very large randomized trials where we actually look at the ability of the vaccine to prevent disease. Then there is this sort of strange category called limited, which is limited approval for use. And this has only happened in China and Russia, which is release of the vaccines before they have been tested in phase 3. This has not happened in the United States. And we have 0 approved vaccines right now, although as you'll see at the end of my talk, we're within days of getting a few approved, or at least approved in what's called an emergency use authorization. Let's go to the next slide. So let's talk about what a randomized trial is, which is going to be our main focus. First of all, we have, in this case, 2 treatments, a vaccine and a placebo. In this case, it's really just salt water. Now, why do we bother with salt water? Why not just spare people a shot or whatever way of giving it? Well, it's because after we give the treatment, we want everybody to behave the same way, because the outcome is going to be if they are COVID infected. And we don't want people who think they've gotten the vaccine to be running into more risky areas. And also, we are going to be looking very carefully at adverse effects. And we want both arms of the study people to think that maybe they got the vaccine. We don't want them to report a bad headache or weakness or fatigue because they thought they had one or the other. So anyway, we start with these 2 possible treatments, the vaccine and a placebo. And then next slide, we basically flip a coin. And you go into one group or you go into the other. Next slide. So then one large group gets the vaccine and the other one gets the injection or administration of the salt water. And then, next slide, we follow them forward and we look at all the possible outcomes. In the case of the vaccines we're studying so far, they actually get 2 doses of the vaccine. So 1 month after the first dose, they get a second dose as a booster. Then we simply compare, and here you see the people colored yellow and red. The red represents COVID infection, but we measure many other things. We measure side effects, safety, immune, we do blood tests, we measure immune response. There are lots of other things we measure, but the main outcomes that you're seeing in the news are they COVID infected or not? And when I say COVID infected, I mean infected with COVID and have symptoms. Next slide. So this is some of the news. No, the previous slide. This is some of the news you've seen, and it is just rolling in. November 9th, we got preliminary data from Pfizer saying their vaccine was more than 90% effective. And then just a week later, just 3 days ago, another company, Moderna, said their vaccine was 95% effective. These are just preliminary results. Well, I guess Pfizer was very agitated at that, looked a little bit more effective than theirs. And they actually then reported somewhat final results. That is that their vaccine looks like it's 95% effective. So all of a sudden, in just 1 week, we have reports of 2 vaccines that might be highly effective and in fact, more effective than anybody suspected at the beginning. And we're going to look at the actual data that was reported. Next slide. Before I get to the data, I just have to show you that this is how it's being reported. It's being reported in a press release, not in a paper, not in a scientific report, although these are quasi-scientific. It's very incomplete. So a lot of the questions that you have and that scientists have, like me, are actually not answered right now. So we're going to have to wait. But they'll be submitting more complete data to the FDA for that authorization, which I'll talk to you about. But I'll show you what's in these press releases. So let's go to the next slide. So this is the data for the Pfizer and the Moderna vaccines. And let's look at the sample size first in the first row. You see that just in the past 6 months or so, they have recruited over 43,000 people to be involved in the Pfizer vaccine, which is a global international trial. And I say 43,000 over the age of 18. Actually, in the US, they're enrolling subjects as young as 12. So we are getting some teenagers in these trials. The Moderna vaccine, which is only in the US, is only adults. They have reported only interim results, and they have about 30,000 people. In the next line, you see basically the top line results. That is, how many people had symptomatic COVID. And in the Pfizer study, you see we had 162 cases in the placebo group, 8 in the vaccine group, and that represents a 95% reduction. You'd expect, if it wasn't effective at all, to see 162 in the vaccine group, but we only see 8. And in the Moderna, really the same sort of result. 90 in the placebo group, only 5 in the vaccine group. Maybe the most important result is the next line, which is severe COVID, because mild COVID, we know, doesn't necessarily make you that sick, although we still don't know about long-term effects very well. Severe COVID, for the Pfizer vaccine, 9 in the placebo group, 1 in the vaccine group, and the Moderna, very similar, 11 in the placebo group, 0 in the vaccine group. In terms of safety, we're really looking at very short-term effects right now. So that is, what we're measuring is both safety and efficacy only a few weeks to 2 to maybe 6 weeks after the last immunization. So these are very short term, and we'll talk more about that in a minute. So as you see, what are called severe adverse events, which is defined as anything that prevents routine activity. With Pfizer, we're getting about 2 to 5% of symptoms like fatigue and headache, but all of these are going away, at least as reported. Moderna, a slightly higher frequency, 10% fatigue, enough to disturb your normal activities. And they're also reporting muscle aches, joint pain, headache, and pain at the injection site. In terms of the mix of these groups, about 40% are what they're calling diverse, populations of color, minorities. Again, the Pfizer vaccine is being conducted all over the world. About half are in older age groups, 43% in Pfizer over 56, and 23, a quarter over 65 in Moderna. And an important thing to understand is that both of these studies are going to continue for 2 years. So we're just hearing the immediate results. In particular, the vaccine arms are going to continue for 2 years. What will happen to those in the placebo arm once the vaccine starts to be distributed? That's unclear. Pfizer actually reported that efficacy looked like it was equal in the elderly and that they had milder side effects, which is very interesting. But again, we have a lot more to learn. Next slide. So I'm going to try to just address these questions very, very quickly. And of course, you'll have more questions later. One, why are the studies so large? What's an emergency use authorization, which we'll expect within the next week or two? What does it mean to say a vaccine is safe, and how safe are these? And what's left to learn? Next slide. So why are they so large? Well, we need a lot of cases in the trials to test efficacy and to test safety. Right now, about 1 in 1,000 people are being infected every day in the United States. That's what's going on, and it's increasing. But if we need 160 cases, if we're going to observe them for just a week, that means we need about 160,000 people in the trial. So just in the placebo group. Or we could observe 80,000 for 2 weeks, or 40,000 for 4 weeks, or 20,000 for 8 weeks. You can see that there's a trade-off between the number in the trial and how long we watch them. But these are the sort of numbers we need to get the number of infections. You saw in the trials we had so far, even though we had 30,000, 40,000 enrollees, we only had about 90 to 100 cases total. So you need those sort of numbers to get short-term results, and that's why we need them. We also need them for safety. If we're going to be looking for adverse effects that don't occur, as I showed you before, like 2% of the time, but instead 1 in 5,000 or 10,000, which we're interested in if we're going to be giving this to tens of millions of people, we need a lot of people in these trials. So you can't find something that occurs 1 in 10,000 if you only look at 10,000 people. You need to look at 20, 30, 40, 50,000 people. So in fact, we're not even going to see enough people in the trial to find all these adverse effects, which I'll talk about in a minute. Next slide. What's an emergency use authorization? This is really important to understand. It's approval to give these vaccines to everybody before the full long-term safety assessments, which will occur at about 2 years. So the criteria for this is if the FDA thinks it is, quote, reasonable to believe that the product, quote, may be safe and effective in treating or preventing disease. It does not mean that it's proven. And it obviously can't be proven if we've only been watching for a few weeks or a month. We can't possibly know what will happen in a year or 2 years, even though we're pretty sure that the bad things won't happen. But we're also in the context of an epidemic where bad things are happening right now, and I'll talk about that. So that's what they're going to be applying for. They're going to be applying for mass use because they believe that these have a reasonable safety margin. Next slide. So is the vaccine safe? And I have to say the most important question you have to say is compared to what? Compared to what? 1 in 1,400 Americans have died from COVID in the last 9 months. At the rate of the current epidemic, if this is maintained, about 1 in either 1,000 or 1 in 2,000 will die in the next 6 months. So that is the background against which we have to measure the safety of the vaccines. We're comparing it, and this is obviously an average. This is the average person in the United States If you're younger, your chance of dying, and here the overall fatality rate is 1 in 200. That's again, an average. It's much, much lower for people who are younger and healthier. It is higher for people who are older, that is older than 65, and particularly if they have underlying conditions. So the mortality risk of non-vaccination in the next 6 months at the current rate is about 1 in 1,000 if the current rates continue, or 1 in 2,000 if we can control the epidemic. So when you say, is the vaccine safe? That's one of the benchmarks you might want to use in helping to decide if it's safe enough. Let's go to the next slide. So far, all the short-term side effects are reversible and brief, but we do have to say we do not know yet about more serious long-term side effects. That is, issues with immunity, neurologic complications, even altered immune response to COVID. Those will continue to be monitored both among the trial participants and in the general population once these start to be given. But we will likely learn it first from the people who are already in the trials. That will be the first important signal because we will follow them forward over time. Next slide. We're almost done. What's left to learn after these releases? Well, lots. The frequency of these rare adverse events, that is less than about 1 in 10,000. We will learn about the things that occur late, that are more a function of the actual immunologic effects of the vaccines. We'll learn how long this immunity lasts and in whom. Right now, it's only being measured 14 days and a few weeks longer, maybe a month or two at most, from the last dose. We'll learn about groups where both the benefit and the risk might differ. We'll learn how many people will take this vaccine and how many will take both doses. We'll learn about the logistics of administration. All of you know that this is incredibly challenging. The Pfizer vaccine requires storage at 75 degrees below zero. And we're going to learn a lot more about immunology and safety and delivery and logistics, and a lot more vaccines will come into play as well. Next slide. That's all I have to say as an intro. So thank you for listening to that, and I'll be handing off to Dr. Grant, and I'll look forward to trying to answer any of your questions after all the panelists. Thanks a lot.
Thank you so much, Dr. Goodman. Okay, indeed, we are proceeding to Dr. Grant, and I see you're going to have to take a brief break from answering all those questions in the Q&A, Dr. Grant, but thank you so much for doing that. So take it away, and don't forget to share an experience participating in a study.
All right, I've been in several studies, and one of my funnest studies was a study that was looking at the effect of antibiotics that are included in various different products we receive, and including in toothpaste. So they wanted to look at how that affected what was going on in our stool. So one of my friends was running a study where they gave you a type of toothpaste that had this chemical in it, or they didn't tell you which kind of toothpaste they would use, and you have to give your stool every two times a week, I think it was. So it's quite a intensive study collecting my stool for my friend, but it was a fun one to be a part of. I don't know if it had any clinical benefit, but it was very, yeah. I'm gonna go into what we're talking about today, which is hopefully will have more clinical benefit than that study did. Next slide, please. And I just wanted to show this slide. This I just got off the Johns Hopkins website, looking at the number of cases worldwide. And I think numbers really don't tell the story. You see that there's 56 million people infected, over 1 million people have died. Now we're over, just since yesterday, we went over 250,000 deaths. But I think to me, like individual stories of patients I know who've died is actually more important than actual large numbers. So patients I've seen in the hospital while doing studies for coronavirus that have tried to do everything they can to stay careful, but for whatever circumstances, whether it be their work or their family members' work, or have unfortunately passed away, or living in nursing homes. And so I've gotten a lot of patients over the last 8 months, I guess, of treating patients in studies. So anyhow, next slide. Oh, this is just my outline. I'll talk about some vaccines and also talk about what we're doing at Stanford. Next slide. This is just the kind of the definition, because it's just any preparation. It can be protein or mRNA at this point that is administered to produce or artificially increase immunity to a particular disease. Next slide. And this is the same thing as Steve showed, just in terms of why the numbers don't exactly add up. Some drugs are in both phase 1, phase 2, and phase 3 at the same time. So they're not double counting that 55. Next slide. I'll just go over why things are moving so fast and kind of give some information on that. So normally it can take up to 10 years to find a vaccine. I do a lot of work with HIV, and we haven't found a vaccine for that in 40 years, since 1981, when it was first described. Many vaccines fail. The fastest prior to this was 4 years for the mumps vaccine. So things have been moving very quickly. And you know, in order to develop a vaccine for COVID, people targeted a 12 to 18 month timeline from the beginning of the epidemic in January to the time it's approved by the FDA. So people are doing quite well in meeting that. The ways that we've been able to speed this up is by using novel technologies like RNA. The regulatory process has been sped up in terms of being able to get things moving through phase 1, phase 2, and phase 3. Some of the early research was done both in phase 1 and in non-clinical research occurring at the same time. The other thing is studies have been occurring, just like I showed you in that slide. Both phase 1 and 2 were overlapping, and phase 2 and 3 have been overlapping. The other thing is this Operation Warp Speed has provided a lot of money for people to produce vaccine even before it's approved. So that's why there is already reportedly, I believe, 50 million doses of the Pfizer vaccine available even before it's been FDA approved. To allow this process, you know, the companies have been supported by Operation Warp Speed to reduce their financial risk. Next slide. These are the studies that are in phase 3. There's 11. There is one that's just a non-specific vaccine, BCG, that is in phase 3, but may not be as protective as the one. Dr. Goodman talked about the RNA vaccines that are Moderna and Pfizer. Both those require 2 vaccines, and they require cold chain. These were the sizes. They started in July. Several vaccines that have been developed in China are using inactivated virus. And again, they require 2. And these are vaccines that have been approved for limited use in China. But they don't have phase 3 data. So it's a different process. We're waiting for phase 3 data to be available before we approve them. There's also these they're non-replicating viral vectors. So they use a different virus that's not able to replicate, and use that virus to enter into the human cell. And once entered into the human cell, then the human cell will produce the outside of the coronavirus, and then we'll develop immunity to what we call the spike protein. There's several studies. I'm working on the Johnson & Johnson study, which I'll talk to you a little bit. Some are requiring 1 shot, some require 2 shots. You know, there's a lot of different ones. Some have been used for many years in the past. The Johnson & Johnson one has been in the platform has been used since 2001. It originally was looked into for potentially a vaccine for HIV. It didn't work for that, but it has been approved for Ebola. There's also kind of your traditional vaccines that use a part of the spike protein and inject that, just like the flu vaccine that you get every year. And then they add something in it to make it a stronger response, what we call an adjuvant. Again, those are 2 shots. Next slide. So I'll just talk to you about the ensemble study that we're doing here at Stanford. Next slide. And it's a randomized phase 3 study. It uses what I describe as adenovirus vector, and it delivers then the RNA of the spike protein. So in the same way, you will get the body will then produce the protein, and then the body will produce an immune response. They've shown that the single vaccine produces a good immune response. They've done animal challenge studies, and that protected the animals. And the immune response looked good in phase 1 and phase 2. We started enrollment in September right now, but there was a pause. People have been following. Some people might follow the news very closely. And like with many studies, if there's an adverse event, there's a pause. So we had to pause for about 10 days. We're up to about 10,000 participants. And we're enrolling a lot of different sites around the world, some in South America. We have a site in South Africa and lots in the United States as well. The study involves 5 study visits over 1 year. We just started about a month and a half ago, and we've been enrolling a lot of patients on one of the buildings at Stanford. Next slide. So the way it works, just like Dr. Goodman described, is you need about 100 events to likely show an effect. The areas, if you can enroll patients who are at higher risk of infection based on their lifestyle, it's gonna be easier to show an effect. If you only enrolled patients that were staying at home and being extremely careful and have a household that's not too crowded, then it might be harder. But based on the numbers, it may be up to 4 months before we find an effect in the Johnson & Johnson study. However, they will look at an effect at 20 infections. And you really can tell by the Pfizer and the Moderna study, there was a lot of new infections between DSMB looks, because you saw they probably were also powered for about 100 infections, but you saw their press releases included almost up to 200 infections. They probably would have been able to find a very strong effect if they had looked probably just two or 3 weeks ago. Although the DSMB has their planned meetings based on what they're seeing. So their planned meeting could have occurred 2 weeks earlier, perhaps. But that's just an aside. We're gonna first look at our efficacy when there's 20 infections. So if you saw a very strong effect, you might actually see a significant effect at that stage. Next slide. This is just how, if you were interested in signing up, you would go to this website, ensemblestudy.com. You enter in your information, and what we're doing, and you write your job, you write down your race, ethnicity, you write down your age, so we can try to make sure we have enough older individuals, people who are at high risk from their occupation, as well as a good racial and ethnic mix of participants. We do have a long list of participants, and so if you do, I have gotten emails from participants outside of the website, which is fine. We also have this vaccine website you can put as well. So we really are trying to encourage, as well as the study sponsors, to have a very representative group of participants in our study. Next slide. And that is, those are my slides. So thank you very much for your attention. I look forward to your questions.
All right. Thank you so much, Dr. Grant. Somehow I knew you were going to have a good story about a study you had participated in, and you did not let me down. And I just want to mention that Dr. Grant mentioned the DSMB. So that stands for Data and Safety Monitoring Board. And these are independent entities that review the data and the safety of all trials, including these vaccine trials. So if you have questions about that, please go ahead and submit them. I see people are submitting lots of questions, and it looks like we have our interpretation was down for a minute, but que bueno, it looks like everybody is escuchando en español, so that's good. All right, so now we're going to move to Dr. Vivian Levy, and we are excited to hear from you as well. And don't forget to share a study you've participated in, or just anything related to participating in research. Great. All right, take it away, Dr. Levy.
Thank you, Dr. Goldman, Rosas. Hello, everybody. I'm Vivian Levy. I'm the Chief of Infectious Diseases here at San Mateo Medical Center, the county hospital and clinic safety net for the county. And I can't really think of a research study I've participated in. As an adolescent, I was involved in a salmonella, a typhoid outbreak in Miami. So I was involved in research that way, but I have two cousins that have been involved in vaccine trials. So a shout out to my cousins. Next slide, please. So San Mateo County has a population of approximately 800,000 persons. Asian and Pacific Islanders make up 32%, Latinos about a quarter of our county, and African Americans 3%. You can see on the map our clinic locations. Here I am with Franco, the nurse, in our 7-bed ICU, and there's approximately 10 county clinics. Here's colleagues at the Pescadero Clinic. Next slide, please. San Mateo Medical Center has really been altered by COVID, like most hospitals in the area. And Latinos have borne a disproportionate hit from COVID, with 93% of all of our cases in the at San Mateo Medical Center being in Latinos, and 89% of our hospitalized cases. And we've had 716 positive cases and 175 hospitalizations up to about 10 days ago. Next slide, please. The overrepresentation of Latinos is well described in the US with COVID, with Latinos being more likely to be hospitalized, as well as African Americans and Native Americans. This is really thought to be due to barriers to accessing care that were in existence long before COVID, to housing density, and to the inability to work from home. Next slide, please. The overrepresentation of Latinos is important in California because it's such an important part of our population. California is 40% Latinos, and Latinos make up 52% of the California COVID cases. But you can also see that other states have overrepresentation, with the purple bars being the COVID cases and the gray bars being the number of Latinos. Next slide, please. And I'll just talk a little bit about why it's important to have, for our county, Latinos involved in clinical trials. Historically, the US hasn't done a very good job of involving Latinos in a number of different clinical trials, be they HIV trials or cancer trials. And in 2012, it was thought that just 1% of clinical trial participants were Latino, while they made up 17% of the US population. Next slide, please. Why it's important for your most affected populations to be involved in your research is, for some of the reasons that you've heard my physician colleagues here say, has to do with outcomes of your highest risk persons, but it also has to do with trust in the process. And I think we could make an excellent case that participation in clinical trials and vaccine COVID research will lead to more trust in the research process and will lead to more uptake of the vaccine and will decrease vaccine hesitancy. Next slide, please. So San Mateo Medical Center is a trusted partner in the county, and we would be delighted to continue to work with Stanford, which is a recipient of a National Institute of Health Community Engagement Award, which really works to mobilize underrepresented populations to decrease misinformation through settings like this one, this town hall, and others, and to improve the recruitment of diverse racial and ethnic populations into vaccine and other COVID trials. Next slide, please. So I'm just going to speak very briefly about three large projects that we've been involved with to try to improve research access. The first was begun under the leadership of Messeng, the infectious disease chief at Stanford, who really had, I think, the crucial vision and wisdom to know that outpatient trials were going to be hugely important for COVID, because 80% of COVID is mild and moderate, and really most of the treatment will occur in the clinic setting. The first Stanford trial was an interferon trial, which, while it showed no benefit as far as the clinical outcomes, I think it was hugely important in showing that Latinos could be represented, could be recruited, and could be retained in a treatment trial. For this study, Latinos were approximately 62% of the 120 persons reported, and it was what UPI would describe as a high touch study with a lot of visits. Next slide, please. Another example of a collaboration has to do with the Office of Community Health and working basically with county disease investigators, who are some of the best messengers in our county at reaching hard to reach populations, and interviewing them for facilitators and barriers for getting COVID and being involved in COVID research. Thank you. Next slide, please. And finally, we created an opt-out research consent that was mailed to 80,000 of our patient members that would allow 3 years of opt-out participation in COVID research studies. So not just the vaccine research, but also treatment research, and even some of what we call the long haulers, some people who have continued symptoms after COVID illness. Next slide, please. And finally, I think just to summarize, building vaccine and clinical trial capacity through collaborations with Stanford and your surrounding counties will improve vaccine uptake in the most affected populations, which in our county to date has really been Latinos. Yeah, I think it's key to have instruments and social marketing materials in Spanish from the start, consents and recruitment materials. And I have really been impressed with Stanford's ability to retain our patients in clinical trials. Next slide, please. And finally, I'll make a pitch. This is our CEO, CJ Canopoli. And thus far, San Mateo County Health System has not received direct funding for COVID research, but we'd encourage you all to check out our foundation website and think about donating, not just for the most vulnerable and at-risk persons, but really to better mobilize our society. I think if we can decrease transmission in those most at risk, we will get our students back in school and all of us back to work sooner. So on behalf of everybody here, the nurses, the doctors, our environmental services, all of the workers that make a huge difference. Thank you for your time, and we look forward to your questions.
Wonderful. Thank you so much for sharing, Dr. Levy.
Thank you.
Okay, and our next presenter, we can turn the slides off. Everyone can take a break, and I'm going to invite Dr. Dubeni to share some of his thoughts, and we're really looking forward to hearing from you, and we can take a PowerPoint break, so that's nice.
Yes, thank you, Lisa and others for the opportunity to share with you. As Lisa introduced earlier, I'm from the Mayo Clinic, and it's exciting to share the platform with folks from Stanford. I'll give you a little bit of a tidbit about myself. I was born and raised in Nigeria, came to this country many years ago, many moons ago. And COVID has been quite a transformative experience for all of us. And I have to tell you, as a family doc, my wife is a family doc as well. And when the first round of COVID broke, I was in Philadelphia with my family. My wife works as a lead for home health services for University of Pennsylvania, and I for Mayo Clinic. And one of the things that we saw as a household, because we're all at home dealing with COVID, was that she was handling the home side of COVID. People were discharged from hospital and getting care at home. And I was working to organize to support our communities as they grappled with the effect of COVID. And it became very clear very quickly that, as Dr. Levy said, COVID was affecting the populations or communities of color in a disproportionate way. And what you saw in some of those slides that is worth repeating is that African Americans were dying at twice the rate, more than twice the rate, sometimes much higher. And I can tell you in some of the smaller communities, the rates are 10 times or higher than the white population. It was also clear for many of us who, everybody who has seen the statistics for Native Americans in the reservations in Navajo, the devastation was obvious. And this was obvious. And it seems nothing seems to be able to stem the tide of the impact of this disease in our communities. So Lisa asked, Dr. Goldman Rosas has asked all of us to share experience in research. And I was fortunate, or maybe unfortunate, as Vivian Levy was describing about this SEAL initiative. And we embraced it wholeheartedly because all of us believe in the importance of working with community, not only to make sure that community owns and empowered and trusted to do the work to elevate the community, but also that when we do interventions and develop treatments, that would do so with the community. And we as a scientific community, we have not been very good at doing that. So here it was, NIH was giving us the opportunity to now launch an initiative. The SEAL initiative means Community Engagement Alliance Against COVID-19 Disparities, to work with the community. And I'm so delighted that I'm in this group to share with you. So we went to our first meeting with NIH, and we're told that one of our missions was to enroll people into clinical trials. And some of us panicked. And for me, the guilt kind of set in and said, you know, I can see the news. Many people are panicked about the idea of upset, angry, and mistrusting about the idea of participating in studies, let alone COVID vaccine trials. As you know, many in our communities have experienced, you know, injustice in terms of research. You can think about Tuskegee trials and other things in other communities that have treated people unfairly in the name of research. And because of those things, many people are reticent, if not angry, at even people who try to participate and sell this idea of participating in vaccine trials. So I want to tell a little bit about why I did. Beyond the fact that I was feeling guilty of having to ask people to do something that I wasn't willing to do. And if I wasn't willing to do it, I felt I lacked the moral authority to persuade people to do so. So that was personal. But on a broader level, we met with some of our community members, and she expressed a fear that existed in the community. She paused for a second and said this, you know, as a young child, we went with our grandfather to receive the polio vaccine. We came back home and we celebrated. We stayed in line for a long time, we celebrated. And guess what? Polio is eradicated. And she said, that's a story worth telling. Then she said, what about smallpox? It's eradicated. And let's think about all the many things that vaccines have actually done. And all of a sudden we realized vaccines are in many ways miracle drugs. Yes, they have potential side effects, but have done things that medicine otherwise would struggle to control. At the time we were looking at this, there was no treatment that was effective for people with COVID. There were lots of candidates out there. Some seem to be promising right now. And it was very clear that if we had a chance to beat this, and it seems impossible to do, that vaccines developed very, very quickly would offer the hope for us to do so. So going back to Dr. Goodman's slides, where he talked about the potential number of people who are affected and potential effectiveness of vaccine. So what I'll tell you is that I went into this with some trepidation, because when I said I was going to participate, many people said, why are you going to do that? Yikes, are you crazy? And I said, well, I have to. I have not participated in the study, and that's actually my day job. I lack the moral authority to do so. I knew there might be some risks, potential risks, the unknown, because you're going to flip a coin, and I may get a placebo, I may get sick. But that's important from science. perspective. There was something else that was important, and I think Dr. Grant talked about this. There are a lot of, I work in the area of policy as well, as service member of the U.S. Preventive Services Task Force, and we know with a high level of confidence, then we'll make recommendations for many things that we recommend, whether it's cancer screening, behavioral counseling, and other behavior change, other things that we know work and save lives. We lack the data on people of color. We make these recommendations not knowing with high level of confidence if they're gonna work in our populations. So if the vaccine is gonna work, there's no assumption we can make with confidence that if it's tested only on white people, that it's gonna work on Black people, Latinos, or Native Americans. The only way to be certain is to test this in all population groups. Because when we know it works in all population groups, when we see differences in these population groups, it equips us in science, in medicine, to be prepared to give the right advice, the right expectations, the right confidence of how the vaccine may or may not work in this population. But more importantly, to me, vaccine offered hope. And I wanted to play my part. There was a lot of helplessness that many of us and I felt as we scrambled with this. I worked in the respiratory clinics, looked at some colleagues who came down with COVID, seen many families who died from it. And if vaccines offered hope, I wanted to be a part of that solution. That was the primary reason why I did so. But let me tell a little bit of my experience of participating, because a secondary reason for doing so was that, you know, you do this work and read the papers about the barriers to cancer, to participating in studies. But we don't walk that walk. It's really hard to know what people experience. Why do we lack diversity? I made a call, scrambled, found the number, and Dr. Grant gave the numbers, Dr. Levy gave the numbers to the trials. I didn't have those numbers. I had numbers from NCI. I tried to find out the companies who did these trials and finally tracked them down. I actually spoke to the CEO of the company that I participated in because I was in this NIH program. I called a number. The reception was probably lukewarm from the receptionist, but that was a job, to screen me and make sure that I was serious about participating. I was asked a number of questions. And at that point, actually, this study, and I wouldn't call the company, I was asked not to do so, but this company has stopped enrolling people other than minorities, people from minority groups. So I qualified because I was Black. I also qualified because I worked in healthcare, in which case I'm higher risk. And of course, unfortunately, I have a bad habit of traveling. So that makes me one of those people who would be a higher risk. And so I qualified. You know, other things nearly disqualified me, but I was fortunate, begging to be eligible for the study, and I was eligible. I drove to the center and discovered something else. They did not have this center open, a trial center open in some other communities that would be accessible to populations of color. They were trying to do this, warp speed was rolling out very fast, and I don't think they had time to think through this. I spent about 4 hours, and it's a long time to spend, but I'll tell you why that was important. I spent a lot of time reading the consent form. I fell asleep, I have to tell you. A few times I nodded up and woke up myself, pinched myself to read through it, because they made sure I had to read through the consent form and I had questions, and luckily I didn't have any questions. And then they brought, as Dr. Goodman said, it came out of a freezer. They had to wait for, I think, 25 minutes for this to kind of thaw out enough for them to inject it into me. And I waited another 30 minutes or so for them to observe me to make sure that I didn't have an immediate side effect. I was equipped with a telephone app to monitor myself, a thermometer, and a ruler to go home to measure the side effects that you've heard about. And I did experience some of those. Actually, after the second dose, a month after the first, I had traveled to Philly, and one night I had a little bit of low-grade fever, and I had to take medication to control the symptoms, but it wasn't that bad, and I got over it. And every week or so, I get a call from the doctor to ask me how I was doing. And if I developed COVID, they would have come directly to my home to look after me. And additionally, I was given $100 as incentive for participating, and I'll get another $100 for the second visit that I completed. So in many ways, I do not know if I'm going to have long-term side effects. I don't know if with that flip of a coin, if I got the saline or I got the actual medication. One uncertainty I have is that if I got the saline, would they give me the actual vaccine when it's actually approved for mass use? I don't know yet, right? And if I got the actual vaccination, some of the questions in the chat box, would I get a long-term side effects? I don't know, but that's part of the experiment. That's part of what we have to do. And everything that we know work in medicine has to be tested in some form. And the best way to test it is through this randomization process where you flip a coin to determine whether you get one treatment or different treatment and compare the two different treatments in terms of the outcome. In this case, COVID infection, severe infection, hospitalization, so death from COVID, to see to what extent is the vaccine preventing COVID from taking hold on somebody and potentially killing somebody. So in this case, it seems very promising, early results, that about 90% of people who, over 90% of people who get this vaccine from Pfizer and Moderna will perhaps be protected, which is huge. That is huge. And I hope that is true. In my heart of hearts, I pray it is true. So what I would offer to you is that if you have questions about participating in clinical trials, I hope you will ask questions of people who have the knowledge to answer those questions. There are lots of places to find information about clinical trials. And you can also talk to a trial center to ask about the why you shouldn't participate in clinical trials. You can ask about the potential side effects or risks of participating and about what they will do if you have side effects or develop complications. But for the most part, the best advances or all the advances that we've had in medicine have come out of experiments, because it's through those experiments that we've learned how best to treat people, what treatments work for people, but also what works for some people, what doesn't work for other people. And without that knowledge, we would not have made the advances that we've made. So I hope that you consider participating in clinical trials, and if not, encourage people around you to consider it. But I also want to offer something else before I stop, which is that when the vaccines come out, I hope you consider getting the vaccine, because it's really important for us to control and contain COVID-19. It's been a long journey for all of us, and containing COVID-19 is really important. And because you know it's still in some ways a rapid experiment, what I hope you consider and do as well is that getting the vaccine doesn't mean you should let your guard down. You should continue to do the same things we were doing to protect ourselves from getting COVID, using a mask, washing your hands, keeping that distance from people, and don't begin to do that high five with others that you don't know or you haven't been around. So that's with family. And if you're exposed to COVID in some circumstance, either at work or at home, you'll isolate yourself and ensure that you're as safe before you begin to mix with other people. Keep in mind that COVID doesn't cause symptoms in some people, so you don't know who has COVID. And you can't assume that because somebody looks well, that that person is COVID-free. So Dr. Rosas and your team, Dr. Goodman, thank you so much for the chance to share with you.
We are so appreciative of you joining us, Dr. Dubeni. Thank you so much for sharing your experience. So thank you to everyone who has been submitting your questions. The first question I'm going to ask actually relates to something that Dr. Dubeni brought up. So you said that you weren't sure yet if you got the placebo, if you would be able to get the vaccine afterwards. So we had a couple of questions, more than a couple of questions, I think, submitted. And this could be for any of our panelists, really. And Dr. Grant, maybe you can answer this for your own trial. If somebody got the placebo, will they be eligible to get the vaccine perhaps as a priority once it's shown to be effective, if it is shown to be effective? And similarly, when vaccines are approved and you're in a trial, will you be able to get the approved vaccine if you're in the trial? So Dr. Grant, do you want to take a stab at that first?
The second question is way more simple than the first question. And the answer is absolutely yes. So when you sign a consent form, one of the important things is it talks about alternatives. You know, what are your alternative treatments? And when that alternative changes significantly, you'll be required, the study team will be required to provide you information on their alternative. And so absolutely, when a medication becomes FDA approved, you will have access and be able to and encouraged to go get vaccinated. So that is, there's a bit of a race for all the studies to get their data before phase 3 data, before FDA approval. EU, as people know, emergency use authorization will be relatively limited, so it won't affect the trial design. The second question, the first question you have is a question that's very important, and I don't think any study has guaranteed their participants priority access to, I've done studies where that is an issue, and we have said, you know, we are gonna allow people to get the drug outside of the study. But I think that's always a big issue in communities is, are you gonna have access to the drug if it's approved, or am I being quote unquote a guinea pig? That was a big issue in HIV studies in the developing world. And to me, there probably should be some public advocacy around this question, that people who were willing to be early study participants, you certainly will be unblinded, that's without a doubt, and be told whether you got the placebo or the active vaccine. But whether you will have priority access to the vaccine when it's FDA approved is an important question that I don't think anyone has answered, but I think is, in my mind, fair for participants. And you know, it is going to be related. There's going to be a big push. Maybe, Jake, you can talk about that, or Dr. Goodman, because I think that's an interesting question.
Mm-hmm. I think Steve is ready to chime in.
I can
go ahead, Dr. Goodman. Go ahead.
Okay, just very briefly. I think I've been involved in some ethics conversations around this, and the way the difference is being split is that people who are in the trials, who are at jump to the head of the line within their own risk group. But the general notion that a person who is in a trial should get a priority over someone at particularly high risk, whether it be a frontline healthcare worker or a frontline worker of any kind, or someone who's at high risk for adverse events, that's generally not thought to be the best plan. But if that person is at higher risk or is in the priority list, they should go to the head of the line within that class of people. So that way they can get some priority. But I don't think we would want a healthy 35-year-old who might be in the trial to get higher priority than a 65-year-old who's frontline worker of any kind, or who is at high risk for either exposure or adverse events. So this has not been totally settled, as we all know. And I think that this is one of the many issues we're going to have to work out as a country and as public health professionals.
Yes, thank you so much, Dr. Goodman. And I think on our town hall in January, we'll be talking about some of these ethical issues related to vaccine readiness or rollout and how we're going to do that. So we'll dive in. There was a great question submitted for Dr. Levy, a question about financial impacts to think about when considering participating in a trial. So Dr. Levy, you talked about the disproportionate burden among Latinos in San Mateo County, and Dr. Dubeni, you talked about the disproportionate burden among many different populations of color, including African Americans and Native Americans. So for participation in clinical trials, the question was, for these groups that might have less access to work from home situations, and then managing the side effects of the trial vaccine that might hinder them from going to work or performing well in their job, would this be something someone should consider when thinking about participating in a trial?
Sure, maybe I'll start and then pass the baton to Dr. Dubeni. I think that the logistical barriers of trial participation, transportation, and frequency of visits are really important for investigators to take into consideration, as are making materials that are understandable and that allow the opportunity for populations to ask questions for clarification. I know that there is stipend and reimbursement, incentive, reimbursement is, or stipend for participation visits. It varies by study. But I would actually like to hear maybe a little bit from Phil as far as how Stanford did such a good job with their outpatient trial and for lambda interferon. If it was, I know that you have bilingual, bicultural physicians as investigators, which I think is some of it, to really allow people to make appropriate expectations of time commitments, or if there was transportation help. Yeah, and I think having, the vaccines are quite well tolerated, so really should not affect too much people's ability to work. But I think the ability to get to study visits is, if you have to be there in person, is really an important consideration.
That's a great point, Dr. Levy. So, and maybe Dr. Dubeni, you could talk a little bit about your own experience, and if you could imagine if you weren't able to work from home, although I know you have to go to the clinic, how participation in the trial might have impacted your job.
Yeah, and that's a very important barrier for many folks, especially if you're working two jobs and you're living from paycheck to paycheck, you don't have time off work. We take that liberty for granted because we can work with a computer. When I have my laptop, that's my work. So it's in my experience, in this study, it required driving to the center. That's time. You need to have transportation to get there. I spent 4 hours in the first visit. So that's 4 hours of time that you have to devote to it. And it could be longer depending on what you need. And this can be a substantial barrier for some people. I don't think the compensation of $100 is enough to make up for potential income loss. But I believe that we can do better. And I think it's one of the points that we are trying to make on the SEAL initiative, that for us to be able to enable people to participate, if our goal is to allow people to participate in these trials, we have to be able to address those barriers. And some of the ways we do this is through navigators, community health workers, providing other types of support. If they have a job that is a constraint, but they're interested in participating, maybe negotiating that time off so they can participate. Some employers are understanding and willing to do that. But I don't think the side effects per se, as Dr. Goodman presented, were severe enough to be a barrier to continuing to work. And sometimes you need to take some Motrin, you feel some soreness in your arm. You do that with tetanus vaccine, and that's not unusual, but that wouldn't be the barrier. But I do think that as a society, and that's my plea, as a society, we actually need to do more to make it more accessible. We have the same structural barriers, if not more, for trial participation as we do with healthcare in general. And trials are no different. If anything, the barriers are even higher.
Absolutely. So I want to change gears just a little bit. We have a bunch of questions asking about mRNA. So these new vaccines from Moderna and Pfizer, we've heard a lot that they are mRNA vaccines. Dr. Grant, can you tell us a little bit in plain language, what should we know about this? Why are these the new kid on the block? What kinds of adverse effects should we be expecting?
I thought I would stay on mute so I could just not answer that question, but the body, the way vaccines work is by creating an immune response to generally to the outside part of a virus. So that the mRNA works the same way. It just gets to that end result faster. So if one remembers classes you may have taken in biology. When the body makes proteins, it uses mRNA within the body to make those proteins. And those proteins within the cell will then leave the cell, and then the body will make immune response to those proteins. The reason they were so quick to be in the market, there was a lot of interest in this for cancer as well as infection before this came along, but they never had made it to any market, towards any market. But all you would need to know is what is the protein, and you can quickly make mRNA for that. Versus, as people know, for flu, you need to get the chickens, I guess, and their eggs to make the protein. So that's why those protein vaccines are a bit slower. Like Novavax, which is protein, we're just getting started. There's a few protein, and those are kind of like, I guess they're kind of like something is going to work. A protein, we've done it. We've done million strong word, but we've done lots of different vaccines with proteins. Sometimes we throw in what we call an adjuvant, but they're relatively slow processes to develop a vaccine. So in order to make it go fast, people did the mRNA. I'm an optimist, and I think as Dr. Goodman said, a vaccine is just like any other medication. There's a risk and benefit to it. And we do know the risk very well of actually getting infected. And so it's impossible, and I'd say inconceivable, that the vaccine will have a higher risk than actually what we know to be the benefit. We may have some rare side effects that came about, but in my mind, the RNA is long cleared from your body. And again, you only have your immune response to the spike protein. So that may be similar from one vaccine to the other. And like we saw, there may be rare neurologic events, but they are very rare. And so for a condition like polio, which we've eliminated, our tolerance for those risks are almost zero. For coronavirus, I'm not saying we should have a vaccine that's risky, but we probably can have a vaccine that has 1 out of 100,000 rare events. People may feel differently about that, and we'd like to have even less than that, but they're understanding that. And as more and more vaccines get approval, there is phase 4 data, which is post-marketing data. There's a robust system to track vaccine adverse events after they're approved. So that happens with every medication, that you sometimes have rare, unexpected events, even after you put 40 to 60,000 through a clinical trial.
Thank you so much for that answer. I'm going to switch gears again. We have some questions about children. So we've been talking about trials that have tested the vaccine in adults. Will there be separate trials for younger children? When will it be considered safe for children? What is the status of that? Dr. Goodman, do you want to take a stab at that first?
I can only answer half of that, and maybe some of the other panelists know more. As I mentioned, Pfizer is enrolling kids down to age 12. So we are going to know something about younger ages. I think it is unlikely that we will have other large randomized trials for young children for a variety of reasons. But a lot of what we learn from these trials and other trials can be used in kids. The other thing about kids is they are at overall lower risk from adverse effects from being infected, although of course they can infect others. So we would tolerate less risk from the vaccine, both because they're younger and also because they have less, we think, less direct harm from the vaccine. I think it's possible, the most likely outcome, but I don't know what's being planned, and maybe others do, that we would start giving it to younger children in non-randomized studies and watching very carefully, and maybe starting with the older ones and working down. So we'll know down to about age 12. We could then, I could imagine giving it to kids, 11 to 8 in graduated fashion, watching very, very carefully and learning slowly over time. I don't think we're going to see randomized trials of the kinds that you're seeing right now, because if we understand that the vaccines have tremendous effect down to age 12, it's very, very unlikely that they will be qualitatively different in younger kids. Now, as you start to get under 5, that's a different story. And I'll let, I think the pediatric, in fact, ID people would have to answer that, because I was trained as a pediatrician. And one thing I can tell you is that children are not just small adults. So there is a point at which we're going to have to think differently about how we learn about both the necessity to vaccinate the youngest. And remember, as more of the society gets immune through whatever means, through infection or hopefully through vaccine, that protects those among us who we don't vaccinate. That's the herd immunity concept, but herd immunity the right way, which is through vaccination. So what the need will be for the youngest to be vaccinated and what the effect is, I think we're still going to be talking a lot about that and working that out. And it may be going on right now, but I don't know of those conversations.
Do any of the other panelists want to add to that? If not, I'll throw out another question.
No, I think what Dr. Goodman said is worth reinforcing. Mm-hmm. But maybe the audience will not take it the wrong way, which is that I'm an epidemiologist, and we don't need to vaccinate everybody in the country to control this. But it's important that everybody who is eligible, as many people who are eligible as possible, get a vaccine, because if we can get a ring around people who are infected, you can block it. Literally, this is like creating a fortress, a boundary, a barrier against the virus. And the way we'll do that boundary is by vaccinating people. And we've done that before. We vaccinate around an index case, and then it blocks its transmission. So children have petri dishes, and they're going to carry something around. But with the uncertainties about long-term effects, I'm not sure that is going to happen anytime fast. But if we can successfully vaccinate, going back to your question, Dr. Rosas, about as a participant, I didn't sign that consent with an expectation that I was going to be given priority to get a vaccine. And so that's one. But I do think it's really important that we get those who are highest risk, those who are triaged to need to be vaccinated first, and control the disease that way. We don't have to vaccinate every child in the world to do that.
Excellent point. So in speaking about children, it brings up 2 topics that have also been hot topics in the Q&A. So one is asymptomatic infections and the other is long-term safety. So let's talk about asymptomatic infections first. We've seen, it looks like from what we've heard from the Pfizer and Moderna trials, they're not tracking asymptomatic infections. And so there are questions about the study design if they're only tracking symptomatic infections. Are we not missing something? Who wants to take that one? Really, any of you could answer that question.
I just want to mention the symptomatic infections are the primary endpoint. I believe that they are tracking, and it might have to be in a subsample because you can't necessarily track everyone, infections in some as a secondary endpoint. But I don't know exactly the design characteristics of that, whether they're doing a random sample of some lesser number of people to test them. I'm not sure, but I'm pretty sure that infection is a secondary endpoint. Again, I don't know exactly how they're assessing that, but maybe others know.
In the Johnson & Johnson study, patients get an antibody at screening, and that will be, and they're going to get blood drawn later, so we'll be able to see how many people got infection. But the actual during the first year, you get blood drawn your first year, and the second year is more or less just safety. In terms of infection, not surprised, each person will be able, hopefully be able to use a device. If not, we can give a device and they report whether they have symptoms. And if they do have symptoms, they'll get tested for coronavirus. So that's like all studies will be and there's a kind of a CDC defined symptoms that will track, that will kind of lead to being tested for coronavirus. So that, because they probably are checking those antibodies in the same time frame, I expect there will be the information of how many people seroconverted. Although I guess it's going to be hard because they've gotten the vaccine. So it's a good question how we are going to exactly say how many got asymptomatic infection. And people aren't being swabbed randomly either. So I think we can say it's preventing severe infection. We can say it's preventing moderate infection. We can say it's preventing mild infection, all to a certain same level of degree, it appears. How much it's preventing asymptomatic infection, unless you did periodic sampling of patients, which I don't think any study, that's quite difficult to do. I don't think any study is doing that. I don't think it would seem very likely to me to prevent, because oftentimes asymptomatic infection is just, if you do a CAT scan on these folks, they'll have disease too, in terms of they will have abnormalities in their lungs. So to me, it may be, the asymptomatic term may not be exactly right. They may be just low level symptoms that aren't being reported. So I would expect although it's a consideration, I don't think it's a huge consideration if we know that direct question exactly. That's my thought.
Yeah, good. Go ahead, Dr. Levy.
Yeah, I'll just add, I think, to emphasize Phil's point that what has really led to death in the United States is surge and overcoming hospital capacity. So from a public health and resources perspective, it's really the critical, severe infection that we really need to prevent. And then, the moderate and mild that Phil spoke of, from just counting, I think really preventing death.
I guess the question also was a question that's unanswerable at this point. And so I didn't really think about it as like, what role is this going to have in public health? In preventing overall number of infections. And that, to my mind, and again, I'm not an epidemiologist, kind of how many asymptomatic infections are prevented, that will have some role in how this can lead to something we can control as opposed to almost eliminate. And I guess that's really a question that we can't answer right now. I don't know, maybe the epidemiologists want to talk about that.
Yeah, I just want to say, if we're able to prevent symptomatic disease, the fear of asymptomatic spread is much less because it doesn't lead to anything but just a silent infection. So right now we're afraid of asymptomatic spread because it can lead to disease which can harm and kill. But if we show that the vaccine can prevent that, then asymptomatic infection becomes sort of an academic and subclinical, question, and only an issue. Well, not really an issue. It's only an issue for the ability to protect those who are not vaccinated, as Dr. Dumaney was mentioning. So that there is an issue there, but for everybody who's vaccinated, it becomes of no consequence.
Just 30 seconds briefly. So I got on the first visit and the second visit a ton of blood drawn from me. I'm glad I'm not anemic, but also nasal swab. So I hope they did that on everybody, which means that they'll be able to detect through the process of randomization, be able to ascertain if there was some asymptomatic infection case. But if it's effective for severe forms, we hope it's also working for the asymptomatic ones.
Yeah, good point. Yeah, we're doing swabs too.
Mm-hmm.
And I'll just point out, Dr. Grant had a few terms, which if you've been listening to our past town halls, hopefully are not new terms. So talking about antibody testing means we're testing for whether or not you've had the infection in the past. Seroprevalence being a measure of whether or not you've been, in the proportion of the population that's been infected in the past. So I'm going to tease our next topic with the last question. Today, we've been talking about vaccine trials, but there have been a number of questions about when these vaccines are shown to be effective, how will this get rolled out and how much time will it take? There isn't enough time in the evening to talk about all the issues related to that. And our next town hall in January is going to be focused on that. We call it vaccine readiness. So what are we going to be doing as a community, as a society, to get ready to roll out those vaccines in an equitable and ethical fashion? So I'll just take one comment from Dr. Levy since she works in our San Mateo County Medical Center. What have you guys been doing to think about how to roll out the vaccine once it's ready? Or a vaccine once it's ready?
So I think this is so important and clearly needs its own town hall. This is going to be a massive lift, the implementation. I think the Advisory Committee on Immunization Practices has been basically surveying counties for the key populations to focus on. Persons here at San Mateo Medical Center, colleagues have been working on scenarios for vaccine prioritization. But this is, I think, nationally going to take an enormous lift, like Works Progress Administration kind of a lift. This is going to really be an enormous effort.
Yeah, thank you so much. So we'll be talking about that in January. I think I put the time up when we first joined. So I want to take a minute to just thank all the panelists. And Maria, if you can put up our slides again. So thank you so much to each of the panelists for taking the time to talk with us today. I want to thank all of the attendees for your excellent questions and for joining us. I especially want to thank the Office of Community Engagement team for putting this all together. And I'd also like to thank the interpreters and Dr. Kosvari for answering the questions. We really appreciate that. And I think I had one slide on another study that people can get involved in if they are interested. It's not a vaccine trial, it's a home testing trial. Maria, I don't know if you can go back a slide or two. Oh. No, well, you took away the slides. Okay, well, we'll be sending out information. If you registered for the town hall, we'll send out information about with the websites of where you can participate if you're interested. And you can also reach out to us with additional questions. And we hope you'll consider joining us in January. And don't hesitate to give comments in the chat or the Q&A about what you liked or didn't like and things you'd like to see next time. So thank you so much, everybody. We really appreciate it.
Thank you.
Bye.
Bye.
Bye-bye.